Hi everyone, my name is Yi Zhang. I am a research fellow at Dana-Farber Cancer Institute working on computational biology. This video is created for the course to give an introduction of the web tool called RegulumDB, developed by the team from Center for Genomics and Personalized Medicine at Stanford. Another similar useful tool is CapitoReg, developed by Kali's lab at MIT and Broad Institute. Today we will focus on RegulumDB functionalities. Recall that we have looked into GWAS SNPs and their linked SNPs in previous videos. So how do we know which SNP is causal in increasing cancer risk? More general, it is usually a challenging task to characterize the function of those SNPs, especially for SNPs in non-protein coding regions. Fortunately, epigenetic profiling achieved by multiple next-generation sequencing techniques can provide some clue to locate the functional elements in a non-coding genome. The figure here showcases the idea of components of gene regulatory machinery. It is known that DNA molecule is packed by nucleosomes on the chromatin fiber with different level of DNA accessibility at different locations. The enhancer can be bound by transcript factors which can regulate the mRNA transcription level of the gene, sometimes from a distal region. So knowing where the non-coding elements are and whether they harbor SNPs will elucidate on the functional SNPs. With this aim, RegulumDB uses probable regulatory elements to annotate SNPs. The potential regulatory element is usually of high open chromatin, which can be measured by the technique called DNA-seq. The elements can also be bound by DNA binding transcript factors, which can be measured by another technique called ChIP-seq. Next, I will now showcase some examples to look at SNPs using RegulumDB data. Now, this is the main page of RegulumDB. Recall that we had one breast cancer associated SNPs in one of the previous GWAS videos, so let's input that RSID and hit submit here. So the search result returned one SNP exactly, and one of the useful features in RegulumDB is a scoring system. So the 2B here links to the details of the scoring, but you can also go to the help page to check what each score means. For example, 2B implies that there is transcript factor binding at the SNP, and the SNP overlaps with some motif scanning results. The SNP is also within DNase footprinting region and DNase peak, which implies it is within open chromatin. So those are pretty good implications that the SNP is following in some functional elements. Going back to the detailed supporting data for this score, the first figure is an embedded view of genomic profiles around the queried SNP, marked by the red line. Clicking on the figure, you will be able to be brought to UCSC Genome Browser, where you may customize the view with more tracks and various visualizations. The default track here includes DNase1 open chromatin states in multiple cell types, and the color here implicates number of cell types in which the region is open. There is also a summarized view of transcript factor ChIP6 clusters from the Encyclopedia of DNA Elements, a database called ENCODE. Note that ENCODE has been updated often so that there could be some more transcription factor ChIP6 data available nowadays at this SNP, which is not reflected in this track. Following the UCSC Genome Browser embedded view, the table shows all protein binding ChIP6 data collected by this version of RegulumDB. Here, each row is a transcription factor binding site, overlapping the queried SNP. The location column here marks the start and end coordinates of the ChIP6 peak. Bound protein is the transcription factor measured by the ChIP6, and cell type is typically the cell line that ChIP6 experiment is carried out in. From this, we can observe some evidence of factor binding, especially for several important breast cancer TFs, including FOXC1, GATA3, and so on. In fact, the function of this SNP was discussed once in one paper before, where the authors predicted and validated that the SNP can affect FOXC1 binding through disruption of a FOXC1 DNA binding motif. The paper is shown here. The next table shows potential binding motif alterations of nucleotide variants pre-computed by RegulumDB. Different from the FOXC1 paper's claim, here FOXC1 motif was not detected according to RegulumDB, but let's still use the ISG factor to understand the motif table. The method PWM stands for position weight matrix, which is a common representation to show how specific the protein binding is at each position. It is plotted as a so-called motif logo in the PWM column, where the higher the letters are, the more specific ISGF3G is when binding DNA at this location. The red box represents position of the SNP in the detected motif. We can thus infer from this that alteration at this SNP in the red box probably does not affect much on this motif. The next category contains chromatin structure data mainly measured by DNA-seq and FAIR, both targeting open chromatin regions. The results here aligns quite well that the SNP is within open chromatin region robustly in the corresponding breast cancer cell type based on the association or in breast cancer. The next table collects histone modifications from the database called RAMSEE, or Roadmap Epigenetics. RAMSEE measured several types of histone modifications using ChIP6 and segmented the genome into different chromatin states in each cell type, with aid of the algorithm called CRO-HMM. This is a sortable table so that you can observe, for example, in which tissue type or tissue group the chromatin state is mostly classified as enhancer region, into heterochromatin, or into quiescent states.